Thank you, Jan. So good morning and welcome. So my colleagues, Jane Rees-Colburn from Reagan-Udall Foundation and Carl Mandel from TB Alliance and myself were asked this morning to kick off by talking to you about the accomplishments of our various working groups within CPTR today. I think before we get started with those details, I have the privilege of sort of grounding us all again in the mission of CPTR, which I think is a really profound one. I'm also going to spend just a little bit of time talking to you about the structure and organization. We have so many new faces, which is an element that's really exciting for me, that have joined the consortium over the past few years. And so I think it's helpful to give you that grounding in how we're organized so that you can understand how we've outlined the discussion topics for today and how we've organized the agenda. So we have a big challenge that we face. And we were kicked off in 2010 with a very clear mission: to move from the drug combination, drug regimen development paradigm that's explained at the top half of this slide, which is a very, very challenging and slow process, to the one that's depicted on the bottom. And we know that there are lots of challenges that we face in order to move from top to bottom of this slide. Some of those challenges are scientific, and we have focused our working groups in the various arms of CPTR to address those specific scientific challenges. I think we can also agree that there have been a lot of strides made in anti-infective drug development and the tools and the science behind how those drugs are developed, but they're not always uniformly applied to the process of TB drug development, and so we're trying to change that as well. There are also large hurdles in the regulatory paradigm when we're talking about combining entirely new regimens very early in clinical trials. And that's really been enabled in recent times through the draft guidance that has been provided by FDA, but I think we all agree there are still some regulatory challenges that we need to work on together. The important point, and the reason that CPTR was born, is that there's no single organization, no single industry partner, no single regulatory body, no single nonprofit that can solve this problem by themselves. And that is why CPTR came together and was launched in 2010 to address this problem and make this possible. And so a reminder of our mission: it's really to accelerate the development of an entirely new, much safer, highly effective regimen for TB. And the part of this sentence that I want to emphasize is by enabling the early testing of drug combinations. And I think we have some progress to be made there, and this is really what certainly the working groups within the Regulatory Science Consortium are focused on, defining those early combination possibilities and paving the way for regulatory review of those early combinations. So just a bit about CPTR structure. The Regulatory Science Consortium is led by the Critical Path Institute, whom I'm representing today. The Research Resources Group within CPTR, led by the Bill and Melinda Gates Foundation, and most recently joining Reagan-Udall Foundation as well. And then the Drug Development Coalition, which Carl Mandel will talk to you, led by TB Alliance. So two of these arms within CPTR, the Regulatory Science Consortium and the Research Resources Group, have very specific working groups that have been established to address those gaps that I mentioned earlier in my talk. I'll talk to you in depth about what we're doing in regulatory sciences, but do want to mention that the Research Resources Group has several working groups as well. I know Jane will talk a bit about these. So the Clinical Trials Infrastructure Working Group, Global Regulatory Pathways, Stakeholder Community Engagement, and Access and Appropriate Use. So just briefly want to mention that we have a leadership body that is helping to guide the focus and mission of CPTR and help us to tackle the toughest challenges that this consortium is facing, and that's led by our advisory panel, which is made up of senior leaders across all sectors. And this will be the first meeting where we will be able to convene those leaders on the advisory panel, and that will happen at the end of day 2 in tomorrow's session. So I want to especially welcome those members of our advisory panel and the delegates that you've sent for this meeting. So a little more detail about the Regulatory Sciences Consortium, which is led by the Institute. I want to start by just reminding the group that we have our own coordinating committee, which is the regulatory or governance body for our specific arm of CPTR. And it's important to mention this because we are really guided by a legal framework that allows the partners within the Regulatory Sciences Group to confidently share ideas and information and even pre-competitive data where that's appropriate. And I think we've finally gotten to the spot in a couple of our working groups where that's going to be especially important. So we're going to be reminding our partners that we really need to draw that data from you and hopefully share the value proposition of what we can accomplish in doing so. These are the partners that have signed that agreement and are members of our coordinating committee and of our working groups. So we have our government and regulatory agencies, our industry members, and also nonprofit sectors within regulatory science who have all signed on and agreed to share this information, readily and as needed by the different working groups. So I know that the title can be confusing. People often ask me, Regulatory Science Consortium, what does that mean? I think we view our role in accelerating the combination drug development process really in 3 large categories. Our aim is to identify the tools and methods that can bring most value to selecting those early combination partners and bringing them through the process. It's really our role to drive and to reach scientific consensus on selecting those tools through sharing of the critical expertise of all of you in the room, as well as information and data that's needed to make really good decisions about the recommendations on those tools that we make. And then, where it's appropriate for different working groups or different programs, to proceed towards regulatory qualification. So again, where it's needed and where it's appropriate, and we have a couple activities in that area that I'll mention shortly. This is simply to share with you the structure of the various working groups within the Regulatory Sciences Consortium. We have 5 main groups. So our TB data standards and integration team, biomarkers and clinical endpoints, modeling and simulation, preclinical, clinical sciences. All of those groups have connectivity to our regulatory partners, and we are constantly asking for feedback and input about the decision and the work that we're engaging in. We have another working group called the Health Authority Submissions Team, which is led by Critical Path Institute's regulatory lead, as well as several regulatory experts from our industry partners. And that team is specifically chaired with the role of guiding the qualification process where the teams engage in those efforts. So a bit about the key accomplishments, and I'm very proud to be up here talking about this because for the past 3 years we've been working very hard towards these various milestones. So the first that I would really like to pay some attention to is the launch of the TB Data Standard version 1.0, which is now published on the CDISC website. And as Jan says, this is not a very glamorous body of work, but it's an incredibly important one when you think about the power of a fully vetted data standard. It allows you to collect data, right, in an organized fashion. It allows you to pool those data where necessary and leverage the power of multiple trial data sets. And when you think about the work that's going to be accomplished, or we would like to accomplish within the modeling and simulation field, for example, having data collected by a common standard and pooled is going to be incredibly important in driving those types of efforts. Secondly, the biomarker clinical endpoint team has selected liquid culture as a predictive or prognostic biomarker for clinical outcome. We are submitting our letter of intent to FDA this week, so kudos to that team for all the hard work and effort that they've put there. And the goal really here is to use liquid culture as a baseline now, as a comparator, as newer biomarkers for efficacy come forward. This will be our way to compare it to a gold standard. The next working group, preclinical and clinical sciences, has really been focused, laser-like focused on really vetting the best preclinical efficacy models that allow you to select those early and best combination partners to get to the end game of CPTR's mission. To that end, we have identified the in vitro hollow fiber tool as being potentially very useful for that decision-making process, and we are working on developing a context of use statement that could support a potential submission for qualification for that tool. And I would like to end lastly on the modeling and simulation effort. This working group was initially developed to focus on the development of a TB disease progression model to enable clinical trial simulation. Still remains the core focus of this team, but this is a great example of how this particular working group has thought very carefully about the science and advancements in the field and are really expanding our work scope to take a more quantitative pharmacology, translational pharmacology approach, starting at the preclinical phase, leveraging data from models such as hollow fiber, to really build and inform the best trial simulation tools to help industry partners make good decisions about combination drug development. Also along this paradigm will be the development of a physiologically based PK model, which will help us to understand drug distribution in the inflamed and infective lungs. So put together, these tools are really, really powerful, and hopefully you can see how we've grown as a team and how these working groups have advanced over time. So I will end my portion on a snapshot showing you where we are in time today at our workshop. And our milestone horizon between now and the next meeting. And I don't want to go into all the details of the various major milestones for each individual working group, but really want to end with this theme, that we've got a lot going on in the coming year, and we need all of you to help guide us, give us feedback, be part of the process, challenge us when we need to be challenged. That's why we've come together for what is truly a working meeting rather than a conference. And so we will have breakout sessions that will happen in the afternoon today. So as you've heard things that interest you or things you'd like to challenge, I would just invite you to attend those specific working groups this afternoon. So with that, research resources.